like to thank the organizers for inviting me here, giving me the opportunity to speak about our biologics LIMS system, which I think will really change the way that we're going to do drug discovery at Amgen. Let's see. So at Amgen, we're committed to unlocking the potential of biology for patients suffering from serious illness by discovering, developing, and delivering innovative human therapeutics. And by the end of this, I hope to convince you that informatics is a strategic enabler to drive innovation and business capabilities for drug discovery, and ultimately to discover and develop the next generation of human therapeutics. So before I move into that, I would first like to give a little bit of the history of the biotechnology industry and really do it in a perspective of how the informatics needs have evolved in the industry. So in the early days of the industry, we were really making things that were just simple recombinant human proteins. So the big challenge here was to discover a protein that could have some type of therapeutic effect and then figure out how to make it. And actually figuring out how to make it was one of the biggest challenges. This is the early days of recombinant DNA technology, and you had to find a way to actually turn this into a potential drug. However, the informatics needs for this was not that great because it was a fairly linear process. You identify a human protein that could be a therapeutic, and you figure out how to make it, and you deliver it to the clinic. So that was really the first wave that really brought forth the biotech industry. The next wave was really to make these therapeutics better. So one of the problems with many of these is that, for instance, that the time in circulation was very short. So we had to find ways to engineer these and actually make them better. So for instance, you might have a drug that had to be injected every single day, and you could engineer it to make it better so the patient might only have to inject it once a month. So that was really the next wave, was making these human proteins better therapeutics. The third wave, though, was where things really changed, and that's this example here. So in this case, the human protein is no longer the drug itself. It's actually the target. So for instance, you might have a human protein that's causing an aberrant problem, and you want to block it and stop it. So the key here is that you have to develop an entirely novel new protein that binds to and activates the target human protein. And this requires a whole new setup to where you're actually doing high throughput screening, looking through tens of thousands to millions of different protein molecules to find the right one that binds and blocks your target. So at this point here, the informatics needs has actually gone up substantially. However, it's still something that can be managed with spot applications. You can do it. But the next wave was when things became even more complicated. And this is kind of where we stand right now, where the pipeline is full of molecules like this. There's like 60 therapeutic antibodies in the clinic and quite a few on the market. But when you go to this stage here, they're just entering the market now, and there's a whole bunch in the pipeline. And these are much more complicated because you have the two human proteins here, which are the target, but they're two different proteins. So you first have to make those. Then you have to find the blocking protein or binding protein, and you have to find two of them, one for each side. So you essentially have to carry out two of these high throughput screens. But it doesn't just stop there. That alone would make things twice as complicated as this. The next problem is you have to figure out how to put them together. And what I've shown here is actually a pretty simple way of just gluing them together, but the reality is there's no set way of doing it. There's over 100 different platforms for actually making a bispecific molecule, and none of them fit all, so you have to figure it out. So it's actually exponentially harder, which means that the data needs have really gone up substantially. So this is just an outline of the basic discovery engine that we use for making biologics. And at the very beginning, as I told you early in the industry, it was a little bit more straightforward. So you identified a human gene that you thought, or human protein that you thought could be a therapeutic. You cloned it. You put it into an organism that could produce this human protein. Could be bacteria, could be mammalian cell culture, and you get it to produce the protein you want. You then have to purify it because that organism not only makes the human protein, it makes all of its own, so you have to purify away all the junk. When you do that, you then have to do the analytics to make sure you've made what you want. And many of these proteins are complicated. They can be up to 10,000 atoms arranged in a very specific way. And so you have to have some pretty sophisticated analytics to make sure you're actually making the right thing. So this is actually a whole host of tests. After that, if everything looks good, you can go ahead and do an activity assay, make sure it has the activity you want. If the activity is good, you can then put it into an animal system, make sure, again, it's looking good. At that point, you could advance your molecule to the clinic. So that was the very early days. Later on, as I mentioned, we went into engineering proteins, and so we actually had to add this top part where you're trying to add parts to the protein to make it better. For instance, to make it stay in circulation longer so the patients don't have to dose so frequently. So that just made the pipeline a bit more complicated. Also, rather than moving just one molecule through, you'd probably be moving a small panel of molecules through. But that third wave is when things became a lot more complicated, because now you still had to make the human protein, which was now the target instead of the therapeutic, and you had to feed this into the discovery platform. And so this is a high throughput platform for identifying proteins that actually bind to the target protein and have some effect on it. The effect could be either to inactivate that protein or to actually activate it. You can look for agonists too. So you go into this discovery platform, try and find things that look positive. You go ahead and take those and you test them for activity. If they look promising, what you usually end up with is a panel of molecules, not just one from the discovery platform, because you're not quite sure which one's really the one you want. You pretty much usually move those immediately into engineering, because the way they come out of this platform is usually not optimized to be a drug. Move them into the engineering platform, and then you have to move that entire panel through this cloning, expression, purification, and analytics process. You then, again, check it for activity. If it looks good, you might move it into in vivo testing. The problem is, typically on the first cycle, you don't necessarily get exactly what you want. You get information about how close you are and what things are working or not. If it's really bad, you might actually take information from these 3 tests and say, oh, you're really off the mark, and you have to go back to the discovery platform and look again. If things are just a little bit off and you think you might be able to just alter the molecule and make it a little bit better, then what you can do is you actually feed back in the engineering module, take that panel, again, expand it, try and fix the problems, make it a better drug, and run it through the process. And you actually have to keep moving through the cycle until you finally get something you're convinced could be a therapeutic molecule, and you can move it then out into development. Then, as I mentioned, kind of with that last phase, it becomes much more complicated because you have to run this discovery engine for 2 totally different molecules. You get them right, and then you have to figure out how to glue them together, and you have to run through this entire thing again and test them together as the bispecific molecule. And so what you can see that this is very cyclic, very interconnected process, and so you're really spinning off a lot of different data at a lot of different points, and so it can actually be very difficult to track what's happening. It's very easy to lose track of what happened at the beginning and where you actually ended up. So to add that to that complexity, there's another issue too. In the case of Amgen Biologics, we're located at 7 different sites around the world. And all of these sites actually interact quite a bit. They are somewhat modular, but there's also a great deal of interaction. So for instance, the San Francisco group may make the target protein. They then hand that target protein off to the group in Canada who does some of the high throughput screening to actually look for the binding molecules. The panel that the Burnaby group then finds, they might hand off to the Thousand Oaks group that then does the engineering to try and make the next molecule, and the Thousand Oaks group might send it back to San Francisco or Boston actually for testing to see how they look. So there's a lot of sharing that's going on here and a lot of data information interchange. And we want to do something that makes that seamless, because the natural tendency is for a site to become somewhat isolated and kind of develop their own systems. So by developing one integrated system that we all use, it really helps with the communication quite a bit. The story is, of course, a bit more complicated than that because we do have a lot of external partners. We have external partners that deliver materials to us. And this can be on a fairly high throughput basis. For instance, DNA molecules, we get tens of thousands of these from our partners every year. So we want to make sure that we have a data system that they can actually directly interact with. So that way we don't have to input all this information by hand when we get it. So we had to set up a system that both would send them information and receive information back from our external partners in a seamless way. In addition, we also have many software packages that are developed both internally and externally. And we had to make sure that the system would be able to interact with those in a seamless way so that, again, we don't have to have humans acting as that interface. So we decided that we really needed to figure out exactly what type of system that we could build that would do that. And we had several foundational needs that we could identify. The first is that we have to have some way of doing sample and container registration. So we needed to know the uniqueness of our samples and the identity of our samples, and we needed to be able to register the containers they were in so we knew where they are and what they were. We needed a way of having complex lineage tracking. So one of the problems is that, for instance, with some of our older data systems, we would know we had compound A and we would know we had compound B, but we wouldn't have great connections between compound A and B. We want to be able to make sure we can track that B came from A. We also needed definable entity transformations. So again, we know now molecule A and B are connected, but we wanted to know how they're connected. What happened to molecule A to make it molecule B? So we needed a way of actually defining those transformations. We also needed flexible data capture, and I'll talk about this a little bit later, but we have a lot of instruments generating a huge amount of different types of data, and we needed a way of capturing all that data in a way that anybody could read. As it stands right now, that could be very complicated because all these instruments generate their own type of data in unique formats. We also needed to make sure that we had good informatics integration. As I mentioned, we have a bunch of different functions across a bunch of different sites, and so we want to make sure that all of these interactions and information exchange between these sites is seamless and it doesn't have to happen by email and spreadsheets. And then finally, we really had to have a method of doing high throughput processing, because that's another thing that's changed in our industry through time. In the early days, it was relatively low throughput moving things through the laboratory, but now with robotics and liquid handling and all the improvements in instrumentation, the output is very high throughput, and we have to have ways of dealing with hundreds of thousands of molecules moving through the pipeline continuously. And so we found that core informatics offered some of the fundamental tools that we would need to build these systems. They gave us entity model registration, controlled vocabulary, sample annotations, defined experiments, high throughput data capture, and very critically, integration with other systems. Because as I mentioned, we have a lot of other systems that we need to be able to connect to our central database. So I'd like to describe a few of the critical elements of the system that we needed. And one of the first is that we had to have three main entity types. So the first tier entity type is the sample. And I think maybe the nomenclature here is a little confusing, because in this case, the sample is not a physical thing. It is really just a concept. So for instance, the sample could be the sequence of a fragment of DNA. It's just the four-letter code strung together that describes what that DNA strand would be like. So that's our sample. We needed a way of identifying it and giving it a unique corporate ID. Also, we needed a way of making sure that it truly is unique. So if somebody goes back and tries to make the same sample again, the system could go back and look and say, no, this has already been made and this is the ID for this particular sample. So the scientist then has that ID. The next critical entity, though, was the substance. So this is the actual material. So in the example I gave, this would actually be the DNA strand which was described by that sequence before. And this is actually connected to the sample because the sample has its corporate ID. The root ID of the actual substance is exactly the same, but the difference is that it gets a dash and then a lot number, since it's actually a substance, it's a real material. And if you go and you make that particular substance again, it just gets another lot number. It says, I'm the exact same sample, new lot number, so we get -2, et cetera. So that's our substances. The final thing we needed was a container. Now, in a very simple model, you could say, okay, well, you could just give the same ID here for your container as you have for your substance. But in our case, we really couldn't. And the reason for that is because we use a lot of pre-barcoded containers. And so those pre-barcodes are not going to necessarily match the assigned root barcode given to our substance. So in this particular case, we had to have a way of connecting them. So for instance, you might have samples in a 96-well plate that has a single barcode on it, and you have 96 different samples in there, each of which is connected to this barcode and then the location it is within the plate. Another common thing is that we often use these little barcoded tubes, and these little 2D barcoded tubes, the 2D barcode is very small at the bottom. And we don't want to put an adhesive label on these tubes because the adhesive label is not very compatible with robotics. You put those in a -80 freezer, the labels start to peel off, and if you use robotics to try and pull it out, it ends up jamming up the works. So our samples are actually stored in these barcoded tubes, and they only have the 2D barcode at the bottom, nothing else to assign them. So we needed a way of connecting that 2D barcode back to the substance. And of course, the substance can be in a bunch of these tubes, so you might actually divide this particular substance up amongst 100 different tubes. So one other critical element that we needed was the experiment. And I look at the experiment really as the glue that ties all of this together. So we have our samples, we have our substances, and we have our containers, but they're not necessarily tied together. The experiment is what ties all of this together. So the critical part of an experiment is that we define exactly what type of thing we're going to do with a substance in this experiment. So each experiment is really customized for the type of process it does. So we actually have a bunch of different types of experiments. Some of that information is already filled out in the experiment. Others, the scientist has to fill in the information about what they're going to do. But it's typically connected to some type of instrument, some type of operation that you're going to do. The scientist fills out the information that they want, what they're going to do with it. They can then scan the container that contains the substance, and so that's directly connected the substance into the experiment. They carry out the operation, they collect the data, and then the data is tied back in to that sample and that operation via the experiment. Now, for an analytical type experiment, that might be the end. You would just do this part here. But if you're actually doing a transformative type of experiment and you're converting the substance into something else, you'll end up with substance 2 over here. And so what this experiment does is it ties substance 1 to substance 2, tells you exactly what you did to it, and it also ties in the data that was generated during that transformation. And so that's why these experiments are very critical for us to make sure that we know exactly what's happening. So they track lineage as well as what has actually happened to that sample. So this is really just a high-level view of our LIMS system. So we call it high throughput biologics. You'll probably hear me talk about it, call it HTB for short. And this is just kind of an overview of how the different samples, substances, and experiments and data are all interconnected. And as I mentioned before, so in the early days, you would really just start down here with the cloning process, right? And so you have your input, your DNA fragments and all that. You're making your clones. You finally come up with your final clone. You're ready to put that into an expression host. Could be a bacteria, mammalian cell culture, et cetera. And you carry out these expression processes. So these are all the different types of experiments that happen with an expression. Some of these you'll actually see are cyclic. So the operations can happen multiple times in multiple ways as you're carrying these things out. They then move into the next stage, which is purification of the molecule. This is another example where you have cyclic processes occurring here. You keep running it through different processes until you get your protein pure enough. Often you're having to do analytics on the side to see where you're at. In the end, you might do your final basic analytics, and then you hand it off for the advanced analytics. And you can see there's a whole suite of different things you can look at for a particular protein product. So that was the simple process, essentially in the early days, which has now been codified by this LIMS system. Thing to keep in mind is that each one of these dots actually defines a fairly complex experiment and has quite a bit of defined data associated with it to define that transformation. Of course, later on, we had to add this discovery module, and this is the case where we're actually screening tens to thousands up to millions of molecules, looking for the ones that actually bind the target rather than just making the human therapeutic. And so you actually have to add this entire section up here. The interesting part, though, is that the discovery module here is actually connected to the purification, expression, and cloning module, and that they feed components into the beginning of the discovery process, because the discovery process requires those target molecules to go into it in order to generate the molecules that bind to them. You then run through this discovery process, and as you run through this process, at the very end, you'll notice you're converting from substances, you end up back to samples, so you're back to the information. So generally, you are producing materials as you're going through the discovery process, but they're not really your end materials. At the end, as I mentioned, you're identifying molecules that have potential to be therapeutics, but we know they're going to need engineering. So essentially, you're generating just information at the end. This information is then fed into this part here, which is actually really the engineering module. And again, that's why this is all samples. So this is just information here. You're basically engineering these initial hits to try and make them better. You then feed them back into this process, and again, you move your way through. And so that's how all of these things had to be really interconnected. The one thing you may notice, though, is that I'm missing some parts here. So I'm missing the part where you're actually testing the activity of the molecule, or you're testing it in vivo. And that's because we don't use our HTB system to track that. We have other systems that are more appropriate for tracking that kind of data. So this is really around the discovery and production of the molecules. So another issue that we've run into is with regards to instrumentation. So this was alluded to earlier in that you have a whole lot of different instruments generating a lot of different data. And these are pictures of just the instruments just in our biologics group. And there's actually over 60 of them. And I'll bet you I didn't even get photos of all of them. And these are just the instruments that generate data that we need to have a way of capturing. So the big problem here is that almost every one of these instruments generates data in its own proprietary format. And so that can be very difficult, because if you try and just upload that proprietary file, it's not that useful. Most end users don't then have the proprietary data software package to read that data on their desktop computer. So they can't read the file. So it's kind of pointless to upload them that way. So one of the things we realized early on is we needed a way of converting the data from all of these instruments into a universal format that then anybody could look up the data. And also, we didn't have to worry about data obsolescence. That way, the data should exist in our system indefinitely. So we really settled on two main data types, which are, of course, really closely related, tabular data and coordinate data. So tabular data is for the instruments that generate relatively low throughput data. It can be represented in a table and displayed as a table for the end user to look at. So that's a large chunk of the instruments generate that type of data. There's a lot of instruments, though, that generate huge data sets. For instance, chromatography instruments. They can have tens or hundreds of thousands of points. And of course, we wouldn't want to present the end user with a table of points. That would be very meaningless. So instead, we represent that as coordinate data as a way of just plotting that out in a 2D type of plot. These are, of course, our preferred data types because these data types are mineable and they're manipulatable. And it's very nice that the end user can then actually take, for instance, these chromatographic plots, and they can zoom in, resize, replot, overlay however they want, because it is a universal format. And they can just do it in Excel if they wish. Some instruments, though, don't put out this data that's compatible with this type, so we end up having to capture images. Just a few instruments are like that. We do have a way of doing that. And then a very, very small number won't do any of these three, and we're forced into capturing reports, but we capture those as PDF reports, so anybody should be able to read them. So along the way, though, we did learn a few things. As I mentioned, we have 7 different research sites for biologics, and The tendency is for sites to kind of develop their own ways of doing things, and that's not very compatible with having a universal type of LIMS system. So we've actually had to work closely with all these sites to see if we could actually settle on agreed upon more universal processes for doing things so that we can make it more compatible with the good LIMS system. So that's actually one of the side benefits of having launched this, corporate-wide, is that we actually ended up with a lot of process consolidation, which really actually helps quite a bit with best practices. So that was one gain, although of course there is some effort in doing that. The other thing we've tried to do is consolidate instruments as much as possible. As you might imagine, it's a fair amount of work to develop the transformation modules to transfer the data. And so quite often there's maybe 5 or 6 different instruments that are really doing the same kind of job. So wherever we could, we actually tried to consolidate that into one type of instrument to help minimize the amount of data parsers that we have to deal with. That also helps too with group interaction. It's a lot easier. We can share scripts and ideas if we're all using the same types of instruments. And of course, we had to develop the parsers to convert all the data. We converted it to universal data format. But then the other issue is we really didn't want the users to have to actually upload each of these files one at a time. That's very painful. So wherever possible, we're trying to develop systems where we can actually do automated data transfers so that the data actually automatically gets pushed into the LIMS system. So another issue we ran into is controlled vocabulary. So I'm not sure with other industries, but I can tell you biologists are absolutely horrible when it comes to having uncontrolled vocabulary. There's, for a single gene, there's often 10 different names, and for each one of those names, there's nicknames. And so if you try and go through the scientific literature in biology, it's very complicated to do any types of searches because you have to basically know all the names and nicknames to try and do a search to get comprehensive information. So one of the things we set out to do was to use very careful and controlled nomenclature to make sure that we really understood exactly what we're dealing with, and that allows us then to actually mine this data in a meaningful way. So we set up for controlled nomenclature. The other problem too is that, as I mentioned, we're kind of in the age now where we're not having simple molecular platforms. We're making these bispecific molecules. They come in a huge number of flavors. So the molecule types are very complex. And so we actually had to build a system which does automatic classification. So you make the DNA construct, the automatic classification system determines what class it is in and assigns it that class. Again, that really helps if you want to mine the data, because now you can ask questions about a specific class of molecule, and you don't have to rely on the nicknames that people are giving these different classes of molecules. Then at a bit more of a detailed level, we also have added controlled annotations. So the classification is really kind of the big picture of what the molecule looks like, but the controlled annotations actually define some of the details. So again, we wanted to talk about these different details in a very controlled and specific way. So again, we can ask questions in a meaningful way and get good results. And then finally, we also have to control the names of the targets. As I mentioned, these things often have many different names, so it can be very confusing what program you're looking at if one person calls a target this and the other person calls it that. So again, we very carefully control target names so that way we can tie our programs in together. One part of the LIMS system that might have been a little bit less obvious, and maybe early on it wasn't completely obvious how we were going to do this, but we wanted to incorporate some type of workflow management. And it really is kind of a natural marriage because your LIMS system to a large degree really is monitoring your workflow because it's following your molecules from the very beginning to the very end. It knows what's happening to them and when it's happening. So what we did was we actually also built the other end, the high end. So we built the top layer, which is basically where you set up the request and you define what types of experiments you want to happen in the request in the very beginning. And so that's your initial part of starting your molecule sets moving through the system. You create a request that kind of sets up the beginning of the workflow. Then the system actually monitors what's happening as your materials are moving through this workflow and reports back to the workflow management. So it's really a very good way of controlling what's happening in your labs and seeing where things are, seeing if they're moving along like they're supposed to, or maybe things are stalling and asking why they're stalling. As a side benefit to all that, you get metrics out of that. And so you can actually look at your process, see what's happening in the process, and determine areas that maybe you could actually do some improvements in. And so that's another advantage of doing this and building this workflow management layer on top. So as I mentioned before, our high throughput biologics system is by no means standalone, and I think this really kind of demonstrates that. So in the first part here, we've actually connected HTB to an internally developed system called BioReg, which is a way of registering large molecules. And this is, as far as I know, reasonably unique system in that it doesn't just register these large molecules by their amino acid sequence or nucleic acid sequence. It actually registers them at the atomic level. So any type of atomic modifications that have actually occurred to these molecules, change them. So for instance, a protein that has an identical sequence but maybe a different disulfide structure actually has a different identity than another molecule with the same amino acid sequence. So this gives us our very specific registration system for, and this here is basically the root number and the lot number for that particular molecule. We have our bit system here, which this was a system that we used to interact with the instruments, so it acts as the interpreter. So basically it takes these proprietary formats, converts them into universal format like a CSV type file or other type of tabular data, and then sends them to HTB. But it also acts as a two-way communicator. So for instance, for our liquid handlers, you might actually create your work list within HTB. The bits module then takes that work list, sends it to the liquid handler. The liquid handler does its task and then sends back information back to HTB to tell HTB what it actually did. And so there's some two-way communications in there with that part of the system. For our engineering module, we use both an internal and an external package. The external package is Genius, which we use for a lot of our molecular biology type work and designing our DNA constructs. And ab initio is an internally developed engineering platform focused mainly on antibody type engineering. And again, this is a two-way communication. So from HTB, you can send stuff up to the engineering platforms, and vice versa. They can also send stuff back to HTB. As I mentioned, controlled vocabulary is very critical, and we have two sources for that. We have the Research Taxonomy Foundation here, which HTB draws from to make sure we're using very unique identifiers, as well as our Research Data Hub, which is our internal classifier system. We, of course, have a fair amount of outsourcing, and we needed ways of interacting with our outsourcing partners, so we had to connect them in. And of course, this is a special type of connection because it's not a broad connection, it's a controlled connection. And this should actually really be a two-way arrow because quite often we're designing things, they go into HTB, the order is then sent to the outsourcing partners to what we need. They make what we need and then send us the information back, which is then directly uploaded into our high throughput biologics system. We also have to have a way of maintaining our inventory, so we use Mosaic for that. So the materials that we're producing are then logged and registered in their containers, and those then go into our inventory system, and that way it's very easy to order up materials. We have a centralized materials management system for that. Users can also have their own personal inventories too, so we have two separate systems for that, personal inventory and then centralized inventory. As I mentioned before, for maintaining assay type data, we use Screener for this. So it's an external software package which is really optimized for containing assay data. And then finally, when we're having this tremendous amount of data sitting here within HTB, what we do is we actually upload that to our data lake and extract it and put it in there. And that makes it then available for our other systems. So we have an internal system for doing queries called Research Gateway, which is a very handy system for asking questions about data, research data. We also have Insight, which is more internally designed. It's more specific for actually assessing samples and comparing them. And then, of course, we have external packages like Spotfire and Tableau. So all of these things can be used to generate queries and reports on the data that sits in our data lake. So this is kind of moving more out into the 30,000-foot view of our research data systems. So we really have 3 layers in this data system. We have the reference data layer. And I've mentioned before that actually HTB draws from several of these. For instance, I mentioned, the biologics identity. It actually both draws to and puts information to that. We have the target identities, controlled vocabulary, and actually it interacts with just about all of these reference systems that sit at the bottom layer. HTB lives in the transactional layer, and as you can see, there's actually quite a few other applications that we have in research that live in this transactional layer. And then finally, all the information that's in HTB is uploaded to our large molecule data hub. Other systems upload to other data hubs here. So all that information is actually stored in one centralized place. And this is actually where the query tools actually pull the information from. So while it's very nice to have this tremendous data lake of information, and it is very useful. It's great to be able to look back at a sample, know what that sample is, know the sample's history, be able to track back. That's a very powerful thing. It's almost essentially like having a very highly organized queryable notebook rather than having every individual scientist have their own notebook. You still have to have those for legal reasons, but essentially almost the same data is there, but it's there in a very queryable format. So that is, of course, tremendously useful in and of itself and would be a reason to have a system like this. But we don't think this is really the only reason to have this type of system. The real big advantage, we think, is the ability to actually mine this data. And of course, right now, we're looking at a situation where we have millions of pieces of data. It's really beyond human comprehension to try and tie all of that data together and really dig into it deep. So what we're really looking at now is using artificial intelligence systems to be able to mine this data. And so, of course, the video card maker, NVIDIA, made this supercomputer that sits in a box the size of a suitcase, which is pretty fantastic. But I think what's even more amazing is that 12 months later, they were able to fit the same computing power into a pizza box-sized computer, which is the one that actually controls the autopilot on the Tesla. So clearly, the hardware is really catching up to the point to where that it could mine these tremendous data lakes. And we want to leverage these as tools in order to help us find new targets and to develop new drugs for these targets. And we really think that this is going to be the way that we generate the next generation of human therapeutics to deal with serious illnesses. So finally, I would like to thank the people who are involved in this project. Philip Tagari, he's our executive sponsor and the head of therapeutic discovery. He was definitely very critical to sponsor us to develop these systems. From the research informatics side, Arun Nair and Randall Chen, Randall's here today, are our executive sponsors for the research informatics side. And I'd also like to call out Jayanthi Subramani, who's actually been leading the research informatics end in developing this platform, and has been a fantastic partner in developing this. There's actually been many people from Research Informatics Core, as well as the Therapeutics Discovery Core teams, as well as Core Informatics, who participated heavily in the development of this project. And there's actually a huge number of other people, mainly scientists, who've actually also contributed to helping us develop this system. Far too many to name. But I'd very much like to thank them for all of their efforts. And I would like to thank all of you for your time and attention. Yeah, so we would definitely like to be a lot further along than we are. So I consider what we have right now to be a bit of patchwork in that you have an instrument, you have to develop a custom parser, you have to find ways of trying to automate that upload to make it as painless as possible for the scientists, so it's not too easy. I think Chromeleon helps a lot with the chromatographic data, although again, that's limited to certain types of instruments. But I think, some of the visions we're seeing, for instance, from Thermo Fisher, where instruments are much better connected, could really be the way to go. And be nice to see if other companies kind of jump on board with this and make this type of integration a lot easier.
You mentioned that you see the experiment and the sort of request as the center, where you start. Are you using a project management tool to do that? Is it a piece of software that starts that process and collates that information?
Right. So we actually have that all within the Core LIMS HTB type system, but we did develop a custom interface for doing that in order to make the management a little bit easier. But ultimately, it really is run through our HTB system. It's not in and of itself really a custom package. It's just really a wrapper that sits on top to help make it a little bit easier to manage it. But it
resource management within that, people, animals, rooms, reagents.
So people, yes. So specific scientists are actually assigned to specific tasks, and it actually keeps date stamps of when these tasks are started, when they're stopped and moving through. As far as individual things like animals and reagents, no, that's the type of stuff that actually goes into the HTB system itself, and that's where that type of information is tracked.
What systems are you using for your in vivo testing, and are they also linked to your HTB?
Right, so the in vivo testing is actually the one that's probably the least mature of the system. So for the most part, the in vivo testing data is the type of data that's really stored right now in reports at this point. It's relatively low throughput. We don't do a tremendous amount of that. Really, almost all the high throughput part is really in generating the molecules and doing the in vitro assays. And so those are where we have the highly automated systems. But for now, right now, the in vitro is probably the least mature part. We'll probably tackle that sometime down the road. Yes?
I have to assume that a company like Amgen has a fairly large and heterogeneous collection of LIMS systems for management of lab information. Do you see in your future a reduction in the number of platforms that you're managing and possibly a convergence toward a single or small set of LIMS systems?
Yeah, absolutely. As a matter of fact, HTB has replaced a whole constellation of separate systems that we did have. So we actually had quite a bit. We're in the process of retiring these systems. There's probably over a half a dozen of them that have been retired. And the more consolidation that we can do, the better. I don't think we'll ultimately end up at the place to where we have a single thing that does it, because some of these things are very sophisticated standalone packages, and it would probably be very difficult to have an all-in-one, but maybe at some point we could. Maybe that's the ultimate goal.
Question, how much of your infrastructure is actually running on a cloud? How much is running on-prem? And are you using, you mentioned these 7 sites, you've got worldwide, are these using as well kind of the cloud to share data somehow?
So that's probably, again, moving a bit into the technical question. I'll do my best to answer that. So we do have all of our information stored on a central server site. High throughput biologics is run through a web interface type interaction. So I'm not sure if that exactly answers your question. If you have more detailed question about that, you'd probably want to talk to Randall Chen, who's here today. All right, okay. Thank you very much.